Okay, good afternoon, everybody. I'm Sharvari Kulkarni and I'm Technical Associate at BDG Lifesciences. First of all, I welcome you to this session on immunology and immunotechnology with virtual lab experiment. And I assure you that this session this webinar will be really productive and informative for you, and at the end of session you will know the basic concepts immunotechnology and one new thing that we have included, and that is virtual lab on Eliza. So first of all, I would like to start with brief introduction of mine. I'm Sharvari and I have 12 years of experience in the field of biotechnology. My area of specializations include immunology, immunotechnology, IPR, bioethics, biosafety, and more importantly, bioinformatics 1 and 2. So on which, as a technical associate, I do conduct trainings, workshops for BDG Life Sciences. I have my research papers published on bioinformatics 1 and 2. Team will put some links on which you can visit our workshops and trainings that are regularly being conducted. Now, I would like to start with presentation. I'll start sharing my screen, and I would like to give brief presentation about the company. Then we will move ahead with actual topic. Yeah, so today we will be talking about immunology and immunotechnology. Let's start in short about the company's introduction. We are BDG Life Sciences with 10 years of excellence in the service of bioinformatics and biotechnology. Our group has been created for students, researchers, and scientists professionals who are related to the field of biotech, biopharma, and bioinformatics. So the company was started in 2010. Our wide area of services include projects, workshops, trainings, online, on-site, webinars, seminars, and conferences. These are some services which are offered by us in the field of bioinformatics, basics and advanced structural bioinformatics, MD simulations, cancer and omics, then drug discovery. As I told you in the beginning, I would like you to visit our website and trainings and services which are offered by us, and take the benefit of services which we offer. So now I would like to start with our actual topic. We will start with immune system. What is immune system? Immune system is basically a defense system that has evolved to protect animals from invading pathogenic microorganisms and cancer. It is able to generate an enormous variety of cells and molecules that are capable of specifically recognizing and eliminating an apparent limitless variety of foreign invaders. So, as our army is the protector of our country, immune system is the defense system which protects animals from pathogens. So why this capacity is there with immune system is because they are able to recognize and eliminate an limitless variety of foreign invaders. How this is done? Immune response can be divided into two related activities: recognition and response. Immune recognition is remarkable for its specificity. It is highly specific. The immune system is able to recognize subtle chemical differences. So, immune recognition is remarkable for its specificity. The immune system is able to recognize subtle chemical differences that distinguish one foreign pathogen from another. Okay, so specificity and recognition. Due to chemical differences, it can distinguish, it has capacity to distinguish one foreign pathogen from other. Now, this type of response is divided into two, effector response and memory response. Now, what is effector and memory? Effector is the response which is produced at the first time of exposure. Memory response is the response which is shown at the second exposure. Okay, so it is kept in the memory. That is why it is called as memory response. Now, there are types of immune system. First, let us see what are the types of immunity. Immunity is basically the state of protection from infectious disease. Okay, so that has to deal with two types, like innate and adaptive. Innate is less specific and adaptive is more specific. The less specific component, that is innate immunity, provides the first line of defense against the infection. Because most of the components of innate immunity are present before the onset of the infection, and they constitute a set of disease-resistant mechanisms that are not specific to particular antigen. But in adaptive immunity, it does not come into play unless and until there is antigenic challenge to the organism. You're getting? So innate immunity is basically inborn. So it is called as innate. It is already present. But then adaptive is after it rises after the antigenic challenge. Adaptive immunity responds to the challenge with high degree of specificity as well as remarkable property of memory. So these are two properties. Innate immunity, it will react in general because it is present in one. Adaptive immunity will react to the challenge with high degree of specificity, okay, and memory. Now, what are the barriers? There are four basic barriers of innate immunity. They are anatomic, physiologic, phagocytic, and inflammatory. So let us see each one by one. Anatomic is skin, your first barrier, right? After that comes mucous membranes. Then physiological are temperature, pH, chemical mediators, and chemical mediators being phagocytic and inflammatory. So this is the basic. Skin means which retards the entry of microorganisms at the first level. Then mucous membranes, they are at the second level, like cilia, mucus, etc. Physiological barriers, temperature, body temperature, pH, acidity in the stomach, it kills some microorganisms. Then chemical mediators like lysozymes, interferon, complements, toll-like receptors. Phagocytic barriers are phagocytosis. In short, I'll tell you what is phagocytosis. Phagocytosis is basically where the phagocytic cells, they engulf the foreign antigen and they form vesicles through which it undergoes the breakdown. And these phagocytic particles, they after presentation, they are killed by immune system. Then inflammatory barriers are there. Yeah, so this was in brief about innate immunity, and now we are going to see what is adaptive immunity. Adaptive immunity is capable of recognizing and selectively eliminating specific foreign microorganisms and molecules. Unlike innate immunity, adaptive immune response are nearly same in all members of species, but this is not the case of adaptive immunity. It varies from person to person, right? So innate is similar, adaptive is different, it is specific. Now, adaptive immunity has very important attributes, four important attributes like antigenic specificity, diversity, immunologic memory, and self-non-self recognition. Specificity, as we saw, it is highly specific to antigen. One specific antigen will have one specific antibody. Diversity meaning it is highly diverse. Memory means it keeps in the memory so that second exposure is heightened immune response. Self non-self recognition is it will differentiate between self antigen and non-self antigen. It has to be there, right? If it is not there, then it leads to disorders like autoimmunity. Now, this adaptive immunity, it requires the cooperation. Two main parties are involved in adaptive immunity: lymphocytes and APC, that is antigen presenting cells. And effective immune response is called when there are two types of cooperation, two major groups of lymphocytes and antigen presenting cells: B lymphocytes and T lymphocytes. In that, B lymphocytes and T lymphocytes, will learn in brief one by one. B lymphocytes, they produce antibodies, they produce memory B cells, they produce plasma B cells or effector B cells. There are two types of cells which are distinguished: memory and plasma. Their main function is to produce antibodies. So see here, this is B cell, this is T cell. B cell, you can see on the membrane of B cell, there are Y-shaped molecules which are present. These are called as antibodies. And T helper cell, on which you have TCR, that is T cell receptor. Along with that, you can see marker CD4 and CD8. Now, wait, I'll just drag this. Yeah, and this is TC cell, where you have TCR along with CD8 markers. So always remember, T helper cell will be CD4++ and TC cell will be CD8++. Now, T lymphocytes, as we have seen in the previous diagram, T cell receptors, they are present on the membrane of T cell. They can recognize antigen which is bound to membrane proteins called as major histocompatibility complex. Unless and until the antigen is bound to MHC or major histocompatibility complex, there will not be reaction of antigen antibody and there will not be effective immune response. What is antigen presentation? Antigen presentation meaning these antigens are presented by certain cells with the help of molecules which are called as MHC. MHC means major histocompatibility complex. There are two types of MHC which are involved, MHC1 and MHC2. Depending upon which type of MHC is there, there are classifications of recognition, T helper and TC. Effect of both the reactions or T helper and MHC is release of cytokines, and these cytokines are also in turn responsible for effective immune response. See here, this is a diagram of cooperation and what are cells are involved in the immune response. You can see in the beginning here, it is internalized antigen digested by cell. Altered cell presents antigen. Then there is class 1 MHC, class 2 MHC. T helper cell recognizes the antigen with class 2 MHC. TC cell recognizes antigen with class 1 MHC. Now these T cell receptors recognize this MHC along with antigen and they become activated. This pathway will be called as activated T helper cell pathway. This reaction will release cytokines. These activated T helper cells, when they release cytokines, they contribute to the activation of B cells on one line and on TC cells on another line. So on TC cell, you can see here TC cell, when they are activated by class 1 MHC, then this type of activation, in turn, the activation of cytokine, they will become activated CTL. CTL means cytotoxic T lymphocytes. These cytotoxic lymphocytes, again, they are capable of killing antigen. One way goes to humoral response. These cytokines, they react or they activate B cells. B cells, as I told you, they are bound with antibody They react with antigen, and on activation with cytokine, at this stage, they will become secreting plasma cells and they will secrete antibodies. These antibodies will bind to antigen and they follow immune response. Is it clear? Diagram is so well explained or so well explanatory that it must be clear. There are three basic things that our T helper cell do. Okay, so it activates cytokines, one way to humoral response and one way to CTL response. Yeah, so if we calculate or if you measure this immune response, how do you measure? It is with the time in terms of days and the concentration of antibodies. You can see here, this time goes, and how does the immune response vary according to it? So first you can see if the antigen A is incorporated, first few days are called as lag phase, into which in the lag phase antibodies to antigen A are prepared. So it takes a time to increase this curve. Can you see here? At around the 14th or 15th day, there is heightened immune response, right? Again, it drops down. Why? Because antibodies will go. Then what will happen? This is primary immune response to antigen A. Then here, if second time exposure is there, you can see this secondary immune response to antigen. There is quick heightened immune response. Why? Because of the property of memory. Memory presence will generate antibodies very quickly. So the second time exposure is heightened immune response. Here you can see. Now, comparatively, this is primary immune response to antigen B. Antigen B again takes some lag and then drops down. Okay. Now, the basic lineage of how our organs of immune system, sorry, cells of the immune system are, how many cells, how many types are there? We start with hematopoietic stem cells. Stem cells meaning cells which will be self-renewing as well as they give rise to different types of cells. So it goes to myeloid and it goes to lymphoid. Myeloid goes to dendritic cell and lymphoid goes to natural killer cell as well as these types of cells. So myeloid progenitor will give rise to granulocyte monocyte progenitor, eosinophil progenitor, basophil progenitor, megakaryocyte. This GM progenitor will give rise to monocyte, neutrophil, eosinophil, basophil, platelets. Okay. And this monocyte in turn again give rise to macrophages. Here in lymphoid progenitor, you have T cell progenitor and B cell progenitor. In that, you have T helper as we discussed, TC. Okay. Then B cell will be B cell as well as dendritic. Now, what are the organs? This is about the cells of immune system. Now, what are the main organs of immune system? They are primary lymphoid organs, which is thymus and bone marrow, and secondary lymphoid organs, lymph node, spleen, malt, and gall. So, we're going to see each in little depth. So, this is the cross section of thymus. This is the cross section of thymus, which shows several lobules separated by connective tissue strands. Okay, can you see here? This is the cross section of thymus, and this is separated by this connective tissue lobules, which are called as trabeculum. Now there are two types or two regions majorly, cortex and medulla. Densely populated cortex you can see by this cells which are known as thymocytes, okay, T cells, immature T cells, which undergoes rapid proliferation. Also here you can see this gray cells, these are nerve cells. Nerve cells are specialized epithelial cells with long membrane extensions, and they are responsible for accommodating the thymocytes. So around 50 thymocytes are accommodated by these nerve cells. Then medulla. Medulla is sparsely populated and it is thought to contain thymocytes that are more mature than cortex. Okay, so this is the main feature of all the organs. Different regions, cells are at different stages and they are involved with different activities. Okay, so in the here you can see in the medulla, medulla is sparsely populated with the thymocytes that are more mature. Also, here in this region, thymocytes, they interact with more different types of cells, like you can see this purple color, cortical epithelial cells, also uh, medullary epithelial cells and interdigitating dendritic cells. These cells are, they are highly specific, or they can produce class 1 and class 2 cells, okay, macrophages. Okay, so this is the diagram of thymus. These are the main regions, as I told: cortex, medulla, trabeculae, capsule is there, then thymocytes are there, cortical epithelial cells are there, interdigitating cells are there, nerve cells is also specialization of, or speciality you can say, of cortical region of thymus. Lymph node. Lymph node is made up of lymphatic system. Basically, this is the lymphatic system. Okay, so this whole lymph, serum, then blood, serum and lymph. These are three fluids according to the presence of blood cells. So the lymph is the part of liquid, or is the part of fluid that is the remainder of interstitial fluid, right? So lymph contains antigen, and this is the lymph node. If you zoom, or if you, for a better view, you can see here, this is the lymph node, and this is the structure of lymph node. There are three main layers of lymph node: cortex, paracortex and medulla, depending upon which types of cells are present in it. Cortex and paracortex and medulla. So these are the regions. You have efferent vesicle, and if you have afferent vessels and efferent vessels, and in that you have capsule. These are the regions where you have B lymphocytes, densely populated B lymphocytes. Then B lymphocytes again, they become germinal centers. So these are some regions of lymph nodes. Now, spleen. Spleen is very important organ. Spleen has two regions, red pulp and white pulp. You can see here, this artery becomes, or it divides into arterioles. And the region which is immediately present besides this arterioles is known as pals, periarteriolar lymphoid sheath, in which you have, pals is the major region which is rich in B cells, okay, that contains lymphoid follicles. Then you have marginal zone. This marginal zone region is there, which is also a specific, and this region of marginal zone are rich in B cells, okay. Then you have this red pulp. Red pulp is the region which is surrounding this sinusoids, okay, and these are erythrocyte filled regions, so that is why they are in red color. So that is why they are known as red pulp. Okay, so this is some basic which I wanted to discuss about immune system. So we've seen cells, we've seen specificity, we have seen two types of immunity: innate and adaptive. Adaptive features of adaptive immunity, then what is cooperation between humoral and adaptive immunity, why it is specific, and then we have gone through some organs and cells of immunity. Now we will see what is the major important concept of antigen and antibody. So this is a typical antigen-antibody interaction. This is antigen. These are antigen binding sites on antibody. Each antigen, you can see here, is like a lock and key. Okay, so antigen is specific to antigen binding site of antibody. This is the structure of antibody. We have two light chains and two heavy chains. As you can see here, two light chains, two heavy chains. Those are having disulfide bonds, and this is called as hinge region. There is variable region, there is constant region of antibody. This is the structure: variable region, constant region. These are antigen binding sites. Antibodies are also called as immunoglobulins, right? Yes. So this is light chain, this is heavy chain, this is hinge region, this is variable region, this is constant region, and this is interchain disulfide bond. We know there are 5 types of antibody, right? IgM, IgG, IgA, IgD, and IgE. Depending upon its specificity in the structure and function also, there are 5 types of antibodies. Yes. Now what happens? These antigens, they on binding to antibody, they trigger immune response. So this is a kind of immune response. This is your pathogen and this is your antigen binding site and this is antibody. So this is where antigen antibody interaction happens. Now, in brief about the interaction which actually takes place between antibody and antigen, there are 4 types of bonds which are involved: hydrogen bond, ionic bond, hydrophobic interactions, and van der Waals interactions. So we are not going to go into much of it, much chemistry of it, but remember these are 4 types of bonds which are involved in any antigen-antibody interaction. Along with the hydrogen bond, there are non-covalent forces which are involved. Now this antigen and antibody interaction is used wisely in wet lab and now in dry lab in order to study the antigen-antibody interaction and design some experiments. These are known as immunotechnology. So immunology, when applied in technology, it becomes immunotechnology. So let us see what are the types of antigen-antibody interactions and how they are involved in designing assays. Yes. So the first reaction is precipitation reaction. So what do we mean by precipitation? We have monoclonal antibody and polyclonal. When this precipitation reaction will occur, whenever antibody and antigen, soluble antigen and antibody, they interact in aqueous solution and they form lattice. And this lattice eventually develops into visual precipitate. When it is possible? It is possible only if the antibody is bivalent. So monovalent antibody will not form precipitate, right? So antibody, if it is bivalent or polyvalent, like this in this diagram, you can see this is antigen. It has only one antibody associated with it. It will not form precipitate. But here, this antigen is involved with antibodies which are polyvalent. So they are forming a precipitate kind of a thing. And that is how they are leading to precipitation reaction. Now, this is how precipitation curve is explained. It is the, if you see here, it is the amount of antibody precipitated versus increasing antigen concentration. So if you see here on X and Y axis, it reveals 3 zones: antibody axis zone, equivalence zone, and antigen axis zone. In the first portion, it is antibody which is present in axis. So antibody is precipitated. Uh, if you see the curve, it goes on increasing. So it will have in the supernatant excess antibody and excess antigen proportion is given. So in the initial portion, you will have antibody excess zone. Now once the antigen added, it will be equivalence zone. At one point is at this peak, you will see the concentration of antigen and antibody which is exactly equal. So this is where the precipitation is occurring at the max. then it drops down because antibody concentration decreases. Now, when antibody concentration decreases, it will be antigen axis, right? So this is how precipitation curve is divided. And this is the peak point where you have maximum precipitation, and that is known as equivalence zone. Now, when these precipitation reactions are performed in gels, then they become acid. So they will yield visible precipitating lines. When antigen and antibody, they diffuse towards one another in agar, and when antibody is incorporated into agar, the antigen diffuses into the antigen antibody containing matrix. Visible line of precipitation will form. You get it? So that means we are incorporating or we are performing this precipitation reaction in gels. Antigen and antibody diffuse. There are different techniques or ways of doing it, but the basic concept remains. You have incorporated antibody into the agar, antigen diffuses into antibody containing matrix, and it gives visible precipitation line. There are two types of this reaction, immunodiffusion reactions, in order to determine the relative concentration of antibodies. They are radial immunodiffusion and double immunodiffusion. Radial immunodiffusion is also called as Mancini method, and double immunodiffusion is also called as Octernally method. Yeah, so this is Octernally method, double diffusion. Here you can see it is agar matrix. Then you have antibody in one veil and you have antigen in another veil. Once both are incorporated, then they travel towards one another. Antibody in one direction, antigen in another direction, and in between this precipitate is formed. So this line which you can see exactly at the center will be like the equivalence zone where the antigen antibody meets each other, right? So this is known as precipitation reaction. Similar concept applied in this technique is radial immunodiffusion, where you have antigen in the center and you have antibody at the corners, at the ring which is present, or it is incorporated in the agar, I'm sorry, antigen in the veil at the center and antibody incorporated in the agar. Now, when antigen diffuses, you can see radially it is diffused, so it is known as radial immunodiffusion. So radially it will be diffusing and it will meet in the agar antibody which is incorporated. Again, the similar concept. So where it is meeting at equivalent concentration, where the place or the where in the place in agar it meets the concentration of antigen which is equivalent to antibody, there will be formation of precipitation ring. So this ring is the like equivalence zone. This is the way it is performed in a plate. Yes. Now the second technique, which is immunoelectrophoresis. Immunoelectrophoresis, basically, as the name itself says, immuno and electrophoresis. It combines electrophoresis and double immunodiffusion. How does it combine? First, you can see here antigen preparation is electrophoresed. This is more clear. Antigen preparation is first electrophoresed and it separates component antigens on the basis of charge, right? After separation of antigens on the basis of charge, antibody is added. This antibody is added to one side of the antigen, and then what will happen? This antibody will diffuse. Okay, so anti-serum or antibody, when it is added, it will diffuse towards the antigens which are separated on the agar. Now, at the place where this antigen and the diffusing antibody will meet, it will form precipitation ring or these curves. These curves they will indicate the antigen antibody interaction. Like this, it is most of the times this immunoelectrophoretic patterns are used to study cancer. You can see here this precipitation curve in 3 different cases: IgG, IgM, and I lambda. Then comes very important agglutination reaction. Agglutination is like, again, it is the interaction between antibody and antigen. In the precipitation reaction, we have seen it results in the precipitate. Here it results in the visible clumping. This clumping reaction is known as agglutination. So antibodies that produce such reactions, we are going to call them as agglutinins. It is the same principle like precipitation reaction. They also depend on the cross-linking of polyvalent antigens. So this agglutination reaction is used in home pregnancy test kits. Okay, how it is used? We have, in what you have contains in the home pregnancy test kit, we have heptane carrier conjugate and we have anti-HCG antibody. Okay, so this is heptane carrier conjugate and this is anti-HCG antibody. What is the test procedure? Test procedure is when you add urine plus anti-HCG and incubate. When you add urine plus anti-HCG and incubate, then what will happen? HCG carrier conjugate will react. This is the basic reaction is between urine, then anti-HCG antibody and this HCG carrier conjugate which is present in the test kit. This is the test procedure. Then what are we going to observe? We will observe for visible clumping. What are the possible reactions? Reaction when not pregnant will be this HCG carrier conjugate plus antibody. Okay, and it will react if the female is not pregnant. Then what will happen? Her urine will not have presence of hormone. So there will be visible clumping, and clumping will give a sign of result on the test strip. Opposite, what will happen if the woman is pregnant? Then what will happen? There will be HCG present in her urine. Now, when you add contents of the test kit, that is antibody and HCG carrier conjugate, there will be reaction between this HCG and antibody, and HCG carrier conjugate will remain as it is. So it will not form clump. And that is how you can detect the pregnancy. If there is visible clumping, woman is not pregnant. And if it is no clumping, the reaction is she is pregnant. Yes, so these are the kits of pregnancy. Then we see what is radio immunoassay. Radio immunoassay is combination of, again, the immunology principles to radiology. It was first developed by Burson and Yalow, and for which they got Nobel Prize. And it was for determining insulin anti-insulin complexes in diabetes. Let us see what is the principle of radio immunoassay. This principle of RIA, basically it involves competitive binding of radiolabeled antigen and unlabeled antigen to high affinity antibody. So what is the procedure here? Is the labeled antigen is mixed with antibody at a concentration that saturates the antigen binding sites of the antibody. You can see here, these are radiolabeled. You can say radiolabeled antigen. These are antibodies. So the labeled antigen is mixed with antibody at a concentration. This concentration will saturate the antigen binding sites of the antibody. Then we add test sample. This test sample is the infected person's or infected serum sample, which is added in quite a large amount. So it is added in sufficiently large amount. What is the purpose? This sufficient large amount should displace the labeled antigen. So what will happen as you add increase amount of unlabeled antigen, it will be displacement of labeled antigen from the binding sites. Now what you measure here at it is the decrease in amount of radiolabeled antigen. So after addition of infected serum or unlabeled antigen, unlabeled antigen will cause labeled antigen to displace. So of course there will be decrease in radioactivity. This decrease in radioactivity will be measured and it will be the sign of concentration. Indirectly it will be converted to antibody concentration, okay, or the presence of detection of antigen. Then comes Western blotting. Western blotting is again one more technique which is responsible for identification of specific protein in a complex mixture of proteins. Okay, so this is widely used. You must have heard for HIV. HIV is detected by means of this Western blotting technique. So this is detection of specific proteins. Protein samples are loaded after SDS treatment. So this is SDS is sodium didosyl sulfate page electrophoresis. These are the separated proteins in the separation gel. Then you have blotting tank where these proteins are transferred to nitrocellulose sheet and then they are transferred to immunostaining for immunostaining of blood, and you transfer you on this nitrocellulose membrane, you transfer your labeled antibody. So this labeled antibody will go and bind to the antigen or proteins which are separated on nitrocellulose membrane, and that will lead to that we will take to visualization with the help of this nitrocellulose membrane. After immunostaining, we will take to the X-ray film, and then we will develop and fix autoradiograph where we can see where the antigen antibody interaction has happened. So antigen binding will be observed by means of these bands. Then comes Eliza, very important technique. Eliza is enzyme linked immunosorbent assay, which is based on antigen antibody interaction. It is done in the 96 well titer plate. It is qualitative and quantitative measurement of component present in liquid sample. So this is the basic principle of ELISA. Step 1 is the coating of antigen on the plate. Step 2 is addition of enzyme linked antibody. So third step is going to antibody to it. So what will happen? Antigen antibody interaction will happen. Okay, so this antigen antibody interaction will cause, this is enzyme linked. So what will happen? This antigen antibody interaction along with enzyme linked antibody, what will happen after addition of substrate? Substrate will cause reaction between enzyme and substrate, enzyme linked antibody and the substrate. So that is how this enzyme substrate reaction will indirectly give us the presence of antibody and concentration of antibody, right? So that is why it is called as qualitative and quantitative measurement of the component. Now there are so now as I have explained the principle, There are three types of ELISA: indirect ELISA, direct ELISA, and sandwich ELISA. Okay, so let me first explain indirect ELISA. Yeah, so indirect ELISA is where antigen coated well is there. We have coated our well with antigen, and you wash it with specific antibody to be measured, right? So once the washing is done with specific antibody, what will happen? Antibody and antigen will be bound. Their interaction will occur. Now washing is done and secondary antibody, which is conjugated to enzyme, will be added. Now, once this secondary antibody conjugated to enzyme will be added, what will happen? If you wash, unbound antibody will be washed off and bound antibody will be there. Once you add color or chromogenic substrate to it, what will happen? This substrate, because of affinity to enzyme, it will go and bind to enzyme. And then this enzyme substrate reaction will be measured or detected by means of color. So this is indirect ELISA. Then sandwich ELISA. In sandwich ELISA, we have antibody coated well in which washing is done and antigen it will add antigen to be measured. Once this antigen antibody reaction has already occurred, we will add in second antibody which is conjugated to enzyme. So what will happen? It will form sandwich kind of a structure. So that is why it is known as sandwich ELISA. So we have one antibody which is coated in the well, then you have antigen, and then you have enzyme conjugated antibody. So this forms enzyme conjugated secondary antibody and primary antibody. So this kind of ELISA is called as sandwich ELISA. Now what will happen in this case? Secondary antibody which you are adding will be conjugated enzyme antibody. Then you add substrate and then color production will be there. So this is sandwich and indirect ELISA. Okay. Now, since I think you have understood the concept of ELISA, now we will go to virtual lab of immunology, which is most interesting part of the module. These are the results, but I would like to perform and show how these results come. Then we will come to do, first let us discuss these two concepts in little bit. And then we will go to virtual lab. So immunoprecipitation is one more concept where you add, or you have in a tube, you have again antigen antibody mixture. Here the antibody is specific to cell extract. Okay. Now what will happen here if you add secondary antibody which is coupled to, unlike enzyme, here we are coupling it to magnetic beads, and simply we are putting magnetic, what you can say, magnet. We are going to apply magnetic force to the tube, so that where there is attraction between magnet and this beads, there will be clump, kind of interaction between them, and they will clump together. Okay. So this also detects the presence. Immunofluorescence is same. Here you are using fluorescent tags instead of enzymes. So here we will be having antibody molecules which are tagged with fluorescent dye or fluorochrome. Okay. Now these immune complexes containing fluorescently labeled antibodies can be detected by color emission, light emission is here. Not a color emission, but light emission. So by means of signal, here is the light. So we've seen different types of signals which are giving us indirect determination or estimation of antigen antibody interaction. Okay, now let me stop with presentation here. Now we will go to virtual lab. I want to just make sure once from the team, is the audibility and video effect is proper? I got a message in between of internet disconnection. Not a problem, right? BDG team, please say yes. Then I will go forward with virtual lab. Okay, anybody from the participant can say yes. Rachna, is everything going well?
Yes, you're
Okay.
perfectly
Yeah,
audible
fantastic.
and visible.
Fantastic. So now we will go to virtual lab. Yeah. So this is the virtual lab of immunology where we are going to conduct Eliza experiment. Very interesting. Everything is mentioned here, and you can have the diagnosis. Presentation is not visible. Audio is fine. But is it a problem with you only, Nimrit? Because I just made it sure not visible. Now is it okay? Everything is visible now? Who was saying there is problem?
Yes, ma'am, it's okay now. It's visible.
It's okay. So the previous part slides you couldn't see?
Yes, ma'am. The Eliza last few slides were not visible.
Last two slides?
Yes, ma'am. But it's okay.
Okay, because I think there was some internet problem in between. It got disconnected and then it stopped presenting, I think. So which slides you missed?
Ma'am, the last 2 or 3 slides, I think so.
Okay, so we will perform this virtual lab and I will come back to those slides. Do you want me to repeat that portion?
No, ma'am, it's okay.
It's okay? Yeah, fine. Yeah, if in the question answer sessions, otherwise if you have any query, I will come back to the slides again and you will get all this as a recorded session, right? Okay.
Sure, ma'am.
Yeah, you can ask in the chat box if you have any problem. So first we'll perform this virtual lab. Fine. You can mute yourself. Now we will perform each step one by one. Now there are certain precautions that you have to always, I don't know whether any of you have performed virtual experiments or not, but you don't have to miss any single click. Okay, so that is the key of operating. Otherwise it's very interesting and it's a good experience as if you are actually doing it in lab, right? So everything manual is introduced. Diagnosis. So first of all, they have given here what is the background of Eliza. Now we know what is the principle and what is the technique. So I will not go again into the detail of it. This is the notebook where every step is mentioned. So it's like when you enter the lab and you maintain whatever steps you have done, that you maintain, it's the same. The system will just note whatever steps have been performed and reason behind it. So it will tell you why all the steps are performed. That is also very good. Then glossary, which is related to the experiment. So all the glossary of ELISA will be mentioned, and then the technical help. So let us do one by one. Let us experience as if we are doing it. First, we will click. Now it's saying that I have already performed this, so I will go to start over and start a fresh experiment. Okay. Yeah. So now this person has entered and he's saying that I need some ELISA sample assess done on the blood samples. So he's just drawn 40 ml of blood from 3 patients, A, B, and C. And he's going to check systemic lupus erythematosus disease. Okay, presence for systemic lupus erythematosus disease, which is one autoimmune disorder. Now he's telling that use the reagents and supplies and test for this SLE. SLE is nothing but this short form of systemic lupus erythematosus disease. And he's saying good luck to us. So let us start. Here rubber gloves are given. You have to perform this protocol. So we'll click on rubber gloves. We have put on our rubber gloves. Then it will say click the centrifuge to obtain the serum and then select serum A to prepare dilutions. So we will click on the centrifuge. Click the blood samples to load them in the centrifuge. Blood samples are loaded. Right? Now we click the centrifuge lead to close and continue. Now our centrifugation is being done. Click the blood samples to remove them from centrifuge. We'll click on the blood samples. Now we'll transfer a sample from each patient into 3 different tubes, okay? And we will prepare dilutions. So first, what we will do, we will click on serum A. Fine. So how to prepare dilutions? We'll click on large volume pipetter to draw 1 ml of serum. Clicked. Now you have to use this up and down buttons in order to make it 1 ml. So I will click on this for 1 ml and then I will click on the tube for serum A. So from serum A, it will transfer into the first tube the required concentration and then it will discard the tip. Now second is we will use up and down button again to set the amount of 1 ml and then click on tube serum A. This is for second dilution. Right? Fantastic. It is discarded the tip. Now the third. So we will click on the small volume pipetter, click on the up and down button for 0.1 ml and then click on serum A. So it will draw 0.1 ml of serum into third tube, discarding the tip. Now what we will do, we have transferred serum sample. Now we will do buffer, additions of buffers. So for that we will click on this buffer. Again add or adjust the pipetter for amounts 1 ml. So what will happen? It will make first dilution. Then we'll click on 1 ml. Then we will click on the first tube, 1:2 tube. Right? Then the next step, use the up and down again in order to make the second dilution. So we will click on this, adjust it to 1:10. For that, we will have to adjust in this pipetter. I'm clicking everything, okay? And you have to adjust it to 9. Once it is adjusted to 9, then I will click on 1:10 tube. Then the next step, use the up and down buttons on the pipetter to state the amount of 9.9 and then click on 1:100. So this is the third dilution. Done. Now we have to click on each and every tube to mix the dilutions. First, second, and third. Now select small volume pipetter that will be used to draw the diluted serum. So we'll click on this, and what will happen? It will start drawing the fluid. First tube, adding dilution to the plate. Right? Discarding the tip. Second tube, adding dilution to the plate. Discarding the tip. Third tube, adding dilution to the plate. Discarding the tip. Then we'll click on serum A. Its job is done. Let us put it away. Similar way, we are going to prepare B. Okay. What is the purpose, guys, you think of this dilution? Dilution is to make results accurate. So false positive and false negative will be avoided. Because if it is having too less amount of antibody, then result will not be positive, right? So, and why we make it with a buffer is because it adjusted or it avoids the fluid from making it too basic or too alkaline, right? Now we'll click on serum B and we will prepare similar dilutions. It is preparing dilutions for B. Now it will not pain us more. So it will do it once it's just shown for A, it will perform for B quickly, okay? Now we will click on the buffer to prepare sample dilutions. With the help of buffer. Done. Now we will click on each tubes to mix the dilutions. Now we'll select small volume pipetter to draw the diluted serum. Click on the first tube. Add dilution to the plate. Then we'll click the tube B to put it away. We are done with B, right? In a similar way, dilutions of C have been prepared. Now we will click the Eliza plate to add the dilutions to the tray. Now you are added to A, B and C, right? Now the dilutions of our serum C also have been added. Now we will click on this antibody, okay, SLE antibody, to add positive control to the plate. We will add positive control. Then we will add negative control, okay? Negative control is buffer. Now we will click on Eliza plate. It will take us to the incubator. Incubation is very, very important step again. You have to incubate the Eliza plate for 37° and 15 minutes because 37 resembles to body temperature and 50 minutes is the time to complete the reaction. So our Eliza plate has been put in the incubator. Now we will set the time for 15 minutes and we'll adjust the temperature to 37. 37, and then click on the green button to start. We are done. Then we are going to click incubator. We'll remove our plate. Click on the plate. We will now remove the liquid from the plate, right? Are you able to connect it to the concept which we have seen? Right. Now we will remove our liquid from the plate. All the washes are done. Now we have to wash with the buffer. Okay, so we will click on the buffer to wash the wells of the plate. Why washing is important? Is because it will remove unbound antibody which is not reacted, right? Now, next step, which is rabbit anti-human antibody. It has to be added in order to continue the reaction, actually perform the reaction. So now we will click on small volume pipetter. It will transfer. And do you understand rabbit human antibody? That means antibody of human is injected into rabbit so that it forms anti-human antibody. So here antigen is like your human antibody. So in every well, anti-human antibody is added. Again, we are going to carry it to the incubator. Load it. Set the timer for 15 minutes. Adjust the temperature to 37 degree. Green button. Yes. Now we will click on incubator. Remove the plate. Click the plate. Remove the liquid from the plate. Again buffer wash. Buffer wash to the remaining wells. Adding buffer. Then you add substrate, horse radish peroxidase substrate, okay, which will be linked to enzyme, isn't it? So there are many steps involved But then you have to be careful and little patient in order to perform each and every step. Click on the timer again for 15 minutes and click on the Eliza plate. I will click on the plate. Right? So this is our result. Now we have to record our results in this check boxes. So you can see here A 1:2, click because it is colored. Second, third, B first and second, C nothing, and this is positive control. Right? Then enter. Fantastic. So here our results have yielded a correct reading. Congratulations, you have completed the Eliza assay correctly. Click here to see your results. So all the steps have been performed correctly, and this shows that interpretation also comes. So this shows that a patient A is likely to have SLE because all the dilutions they show presence. Patient B might have it because both the dilutions. Patient C does not have SLE. This is positive and negative control. Fantastic. So this is the analysis of a result, and you can maintain the result. Now, what happens even if a single click is missed in virtual lab? Like when I was practicing it, I will tell you, this Eliza washing with the buffer was missed out from me. So it gives you cross over here, and in the result, I got false positive results. So it told me that your results are false positive. Here, since we performed all the steps with very patience and step by step, our results got correct. Understood? Yeah. So now I think I'm done with all the explanation part and virtual lab also. Hope this experience was good to you. You understood each and every step perfectly. This is HMMI lab, virtual lab, in which Eliza virtual they have designed. Okay. So like this, some not actually in microbiology, biochemistry, in bioinformatics also, many virtual labs have been designed by Amrita V Lab and our IIT NPTEL, etc. And there is one, what is there? Webster is also there. Some foreign universities are there for virtual labs which are paid. But you can explore for yourself. But unfortunately, immunology have incorporated only this, Eliza, as a simulator. Otherwise, you get virtual lab in terms of documents, animations that you get for this diffusion techniques, precipitation techniques, but actual performance of Eliza, feel of the lab is given by this HMMI. Howard Hughes Medical Institute. So if you want to use this experiment, if you want to teach it to your students, you can go through HMMI Bio Interactive. You can see here HMM Bio Interactive, and other experiments also you can find out if you are keen to find out or explore more experiments in biotechnology. Okay. Yes, so I'm done with the explanation part. Now tell me if you have any doubt. We'll put some time for question and answer session. So yes, so now I will stop with my presentation. Anybody?
I think I understood everything. It was clear, the basics of immunology also, and the virtual lab was also very well explained.
Thank you.
Thank
Thank
you.
you. Most welcome. Most welcome. Anybody has anything to say?
The virtual lab experience was very good and very interesting. Like, this is the first time I've witnessed something like this. So it was very interesting. Like, the lecture was not at all boring and it was really very
Yeah,
clean.
it's Sunday afternoon, so I didn't want to make it boring. I tried my level best to make it productive because it's the sacrifice of sleep, right? Afternoon nap. Fantastic. It is. I'm happy to Ayushi to get your feedback.
Thank you.
Thank you, Nimrit. Yeah. So enjoy. Thanks a lot. It's a great pleasure. And I would like you, again, I would like to focus on the thing that we have such kind of webinars. Next is, thanks a lot if you had great experience, Nessa. So next webinar will be on bioethics, biosafety, which will be hosting by me again on 2nd. So keep on visiting our website. We have different webinars on nominal prices, and you can also enroll for our workshops. We definitely assure best services from our side. Okay. So if you all are okay, then we can end the meeting. BDG team, some assistant is there. Shall I end the meeting? Anybody is there from BDG? Okay, fine. I think we can end the meeting then. Okay.
Okay, thank you so much.
Yeah, thank you so much and welcome for all rest of the workshops, upcoming sessions. Thank you so much. Bye.